So thanks, Fraser, for agreeing to speak today. I met Fraser again in London in December. Fraser is known in the field of layer fMRI, specifically for a couple early application studies, I think under Peter Koch. He has a lot of thoughts and also data on layer fMRI with epilepsy, working at King's College with David Carmichael. And the title is Developing Layer fMRI as a New Biomarker of Epilepsy in Children for Treatment Optimization.
Hey, thanks, Renzo, and thanks everyone for attending. So today I'm just going to talk about the work that David and I have been doing at King's in relation to laminar fMRI and its use in epilepsy. So let's just get started. So we work mainly with childhood epilepsy, and I realise it's not an epilepsy group, so I'll just give a little bit of background to that. So epilepsy is the most common serious neurological condition in children. And it affects approximately 1 in 200 school-age children ages 5 to 19. Okay. So just a little bit of the pathophysiology of epilepsy. In some ways, epilepsy is relatively well understood, and it's broadly characterized by an imbalance between excitatory and inhibitory signaling in the brain, which converts a normally functioning network into a hyperexcitable one. Obviously, there's a lot more to epilepsy than this, but this is the basic mechanism by which seizures occur. But despite this general knowledge of the sort of underlying cause of epilepsy, there's no real cure for epilepsy. The first line of treatment is drugs, anti-seizure medication, and between 30 or 40% of children will not achieve adequate seizure control with current anti-seizure medications. And also another issue with anti-seizure medications, they only aim to address symptoms. So they only address to reduce hyperexcitability, and they don't address the underlying causes of seizures. So in some children with drug-resistant epilepsy, surgery will be an option to try and remove the seizure originating area. But this success rates can be variable and seizures can often return. And a key thing with epilepsy surgery is that surgery is reliant on an accurate localization of the seizure generating zone. If they can't find this, then surgery is not an option, and the seizures will still persist. So this sort of raises an understanding, what's missing in our understanding of epilepsy. Like seizure drugs have been researched and developed for many years now, maybe 100 years, and success rates have not really improved dramatically in the last sort of 50 years. And obviously, surgery rates are variable, and it's a very drastic mechanism to reduce seizures. So one thing that's sort of become evident in the literature now is this disconnect over scales in which we understand epilepsy. And there's different ways you could sort of classify this, sort of micro to macro, this type of thing. But we've sort of defined it here as nano to macro disconnect. And I'm just going to give an example here of how anti-seizure medications are applied and how epilepsy is sort of clinically managed and monitored in humans. So there's lots of so epilepsy is a multi-scale disorder, really, and any one of these scales, so from ion channels, receptor function, hyperexcitable neurons, damaged microcircuits, aberrant connectivity, hypersynchronous networks, they're all involved to create seizures. And of course, what we're dealing with as well, this is have a really very detrimental impact on life quality of children, sort of across the board, social life, family life, general life quality. It's a serious thing. But the main way that drugs are treated in the first line treatment is epilepsy. And all epilepsy drugs, to my knowledge, well, most certainly 95% were developed in animals, mainly rodents. And most of them target ion channels and receptor dysfunction, often sodium channels. But the way that drugs are administered and the way that sort of epilepsy is clinically managed and diagnosed is via macro scale imaging tools, EEG, fMRI, PET, which is a long way in scale from ion channel and receptor function. So, and this has led to a situation where clinicians don't really have any insight into how anti-seizure medications are actually affecting their proposed targets. They can only measure their effects at the macro scale. And then it's almost like a trial and error process of drugs. They'll prescribe one drug. If that doesn't work, they'll prescribe another. And this has led to a situation where there's a lot of variability in the treatment. So you could have one patient that ostensibly has a same type of epilepsy as another, and they're both prescribed the same drug, and it'll work in one patient and not another. So this is a common sort of failing of epilepsy treatments. But going back to this, so now there's this discussion about the role of microcircuits in epilepsy, which is sort of the increasingly considered a bridge between nanoscale structures like ion channels and the macro scale measurement networks that measure connectivity in network. And this is sort of something that we've been thinking where differing scale at the differing pathology at the nanoscale may lead to alterations in microcircuit function, ultimately contributing to seizure generation and propagation across networks. So just a little note on microcircuits, just to provide a bit of definition and a little bit of discussion on that. So microcircuits refer to widely recognized and recurring patterns of neural connectivity on organization within or across brain region. And there's a few examples of this in the brain and nervous system. For instance, a trisynaptic circuit in hippocampal structure and central pattern generators in the nervous system, the spinal column, which are responsible for like repetitive motions like walking and this type of thing. So, but why is understanding these microcircuits important in epilepsy? So this is from a really good paper that we've cited quite a lot in a publication that we're trying to get out. But all seizures appear to originate in local microcircuits and then propagate from that initial ictogenic zone. So these microcircuits appear key in the hippocampus and cortical layers, which we'll come to in a minute. All seizures appear to originally initiate in that area and then spread from there. So, like I say, the idea is that these microcircuits form a bridge between the nano and the macro and the network level seizure activity that we measure with EEG and this type of thing. So some microcircuits implicate in epilepsy. Temporal lobe epilepsy is a very common, probably the most common type of epilepsy. And some of the microcircuits involved in this, like the cell laser hippocampal subfield, like CA1, these are not currently possible to non-invasively image. But cortical layers, well, now we're approaching the spatial resolution and imaging techniques where we can infer activity at layer depth, which correspond to the anatomical position of cortical layers. So just a little note on something that just wanted to make clear about cortical layers in epilepsy. So probably familiar to everybody here, interlaminate, the laminar structure of the cortex, highly connected, and this facilitates the flow of normal neuronal processing, so feed forward and feedback. But somewhat ironically, these high level of recurrent connections, this makes the cortex highly susceptible to abnormal synchronous behavior. In some ways, because it's so connected, any hyperexcitability or any abnormality that's causing this can quickly spread. And there also seems to be something about layers that makes them vulnerable to hyperexcitability. For instance, piriform cortex has 3 layers, and there's studies where it doesn't take much manipulation to make these when they take the slices to make these hyperexcitable. So there's maybe something to do with this layered, or maybe just it's the recurrent structure. But I just thought that was worth mentioning that this highly recurrent structure makes it vulnerable to synchronous activity. It makes the cortex vulnerable. So now I'm just going to talk a little bit about some work from animal studies and humans, where we have specific evidence for cortical layer dysfunction in epilepsy. So different types of epilepsy, and sometimes they're grouped into generalized epilepsy and on focal epilepsy. So generalized epilepsy is characterized by seizures that affect the entire brain. And these are often linked to genetic mutations that affect ion channels and sodium channels. Mutations are especially well known for this. This SCM1A is responsible for some of the more serious types of epilepsy on the spectrum of epilepsy. So this is a paper that we found quite interesting, and this looked at layer activity, a specific form of electrograph activity called spike and wave discharges in a genetic model of absent seizures. So they investigated the mechanisms of underlying the
Fraser,
initiation of, yeah.
sorry, may I interrupt? It seems that not everybody's seeing your screen. I'm
All
seeing
right, okay.
Ravi's nodding. Sorry. Could you just unshare and share again, maybe?
Yeah, sure, yeah.
I apologize.
No problem. Is that any better?
Yes, now working. Thank you.
Ah, great, no problem. Yeah. So this study investigated the mechanisms underlying the initiation and propagation of spike and wave discharges in gears, and spike and wave discharges are a electrographic hallmark of absent seizures. And an absent seizure is when the person or animal undergoing this will sort of switch off, they will go blank. It's not the type of seizure that you might associate with epilepsy. It's more of a switching off type mechanism. And GERS are a type of rat that they induce a genetic mutation which produces these spike and wave discharges in absent seizures. So, they hypothesized that deep layer neurons within somatosensory cortex play a leading role in seizures in generating spike and wave discharges, which are the hallmark of absent seizures. And they examined the firing patterns of neurons in different cortical layers with laminar electrodes and their temporal relationship to electrocartography, the ECOG spike in motor cortex. So just to explain this a little bit. So one way to think about this is that the ECOG is a type of intracranial EEG electrode that lies on the surface of the cortex. And this measures from a relatively, I say, wide area of cortex, something that each sensor will have in the region of around 500,000 neurons. And then once they've determined the broad location of the seizure, so somatosensory cortex, they will use laminar electrodes, which are a type of multi-contact electrode with really high spatial resolution of electrodes on the shaft that goes into the cortex, and this allows them to gain depth measurements that correspond to the cortical layers, basically. So we made these plots from based on this paper. So I'll just run through these. So the x-axis represents the different cortical layers. So they're measured from 3 layer depth, superficial, middle, and deep. And the y-axis is showing the time difference between the action potential occurrences in each layer and the peak of the ECOG signal. So the peak of the ECOG signal is basically the seizure, the height of the seizure. And just to reiterate what the different electrodes measure and how they refer to this plot, basically. So in
Could you
this
tell
plot
us,
suggests
sorry, what's the time scale? We are not talking milliseconds. This is
it's
seconds.
milliseconds, yeah,
Milli
milliseconds.
Thanks.
Yeah, sorry, yeah. And this plot suggests that neurons in the deep layer fire earliest relative to the peak ECOG, followed by superficial layer and then the middle layer. So, and not only do the deep layers appear to fire first, but they also seem to fire the most. So here, this is the ictal firing rates by region. Again, the x-axis are the layers with the motor cortex as like a basic general control. And the Y represents the ictal firing rate, which is the frequency, sorry, it was in hertz, I think, generated by neurons in each region during the ictal period in hertz. Yeah. So here we see the deep layers exhibit the highest ictal firing rate, and it's significantly higher than the other regions measured. So this suggests that deep layers are particularly active during the ictal event and probably playing a crucial role in its generation or propagation. So the next type of epilepsy I'm going to talk about is focal epilepsy. And this is also known as partial epilepsy. And this is a type of epilepsy where seizures begin in a specific area on one side of the brain. And interestingly, for laminar fMRI, which we'll come to a bit more in a minute, this is often comes from damage to the brain. So we'll come back to this in a second. So this is a really great paper from Bourdillon. It's quite really recent, last year. And the interesting one about this paper is because this is done in humans. So there is research in animals in focal epilepsy, which has demonstrated various different findings in regard to which layers initiate and propagate seizures. Some find deep layers, some are found superficial. And this is probably due to the way that they induce seizures. They chemically induce the focal seizures. And obviously, the type of brain in animals, especially rodents, what they seem to use a lot, is very different from a human. But this is why this paper's so sort of so good because it actually deals with humans. So here they wanted to understand how spontaneous human focal seizures start and spread at the level of microcircuits. And they use arrays of electrodes to record the local field potentials and multi-unit neural activities across layers. So they measured 30 seizures from 10 individuals, and there were different types of lesion in the brain. So focal cortical dysplasia, which we'll come to in more depth in a minute because this is the one we're really interested in, mesial temporal lobe, and I think there was one case of a tumor. So as I say, FCD or focal cortical dysplasia is especially interesting for laminar fMRI because this is actual disruption and damage to the neuronal layer, to cortical layers. And this normally happens in development. The cell plates don't migrate properly and they get abnormal cells, balloon cells, and this type of thing. So obviously, this has obvious implications for laminar fMRI because the cortical layers are actually not properly formed. So this was sort of, I think this was a sort of mean across all the different types of lesion. So here, the x-axis cortical layers, and y is normalized current source density. So current source density is a measure derived from local field potentials that indicates the net flow of current into or out of a specific region of the cortex. So positive values, sinks, indicate a net inward flow of current, and this is typically associated with synaptic excitation, postsynaptic currents. And the negative value, sources, indicate a net outward flow of current. So what you're really seeing here is that during the seizure initiation, there is just more current flow in and out of different cortical layers compared to the supragranular or more superficial layers. So again, consistent with the generalized, it's showing that deep cortical layers especially seem especially important in seizure initiation. So now I'm just going to go into the laminar fMRI part of what we're doing. And you know, as has been talked about by everyone in this room will know, so we're aiming to provide a link between cellular or nano and the macro scale. And we want to use laminar fMRI to detect layer-specific functional changes associated with epilepsy. But the two things we need to be certain of, or at least confident of before, to justify its use, really. And one is, can we detect the eleptogenic electrophysiological abnormalities that we've talked about here and in other papers, seen in intracranial EEG or scalp EEG in the laminar fMRI signal? Obviously, it's going to be very important because these could be quite subtle, really. So just a little note on this, just before we present some data some evidence that showed that we probably can. Epileptic activity is characterized by electrical activity at different frequencies. So, for instance, photosensitive epilepsy is associated with increased gamma oscillations, and these occur in the range of 30 to 100 hertz. That's just a little. I've just got a couple of notes for this slide, actually, the next slide. Just bear with me. Yeah, so in context with this, so there's a few papers that lead us to believe we probably can. So Mendoza Holiday, 2024, this paper deals with a spectrolaminar motif. And this showed that there were distinct frequency bands associated with specific depths. High frequency power, likely gamma oscillations, increase from deep to superficial layers, peaking in layers 2 and 3, while alpha beta power shows the opposite trend, peaking in layers 5 and 6. So in this paper, tested this across a few primate species, and they found the same outcome. And this shows that this is preserved across primate species. So the next paper was from Scheringer et al. And they used simultaneous EEG, scalp EEG, and a high-res fMRI, 7T fMRI, to demonstrate distinct relationships between specific EEG frequency bands and activity in different cortical layers. And in a visual attention task, they found gamma band EEG power correlated positively with the Bold signal in superficial layers, while beta power showed a negative correlation with deeper layers, and alpha power with both deep and superficial layers. So, and this is a paper from David, who's in the meeting today, David Carmichael. And this used ECOG and standard fMRI. I think it was 1.5 Tesla. And they investigated the hemodynamic correlates of intracranial interictal epileptic discharges, or IEDs, in 3 patients with focal epilepsy. And in this paper, they were able to map bold changes associated with IEDs, and they revealed both local and distant hemodynamic changes. Okay, so overall, there's specific frequencies associated with epilepsy. People have been able to map these specific layers. They've also been able to map them with EEG, scalp EEG to laminar fMRI signal. And we've also, and the ECOG paper was also able to relate intracranial discharges to Bold signal. So together, these studies suggest that layer-specific epileptic features seen in electrophysiology in EEG may be detectable as layer-specific fMRI responses. So there is some basis to the idea that we will be able to record these subtle changes or not so subtle changes associated with epilepsy. So now I'm just going to talk about what we've been doing, what we plan to do, and sort of where we are with the project. So like I say, we want to use laminar fMRI to detect FCDs, focal cortical dysplasias. And one issue with FCDs is they can be very radiologically subtle and they can occur anywhere in the brain. So a lot of the time they report as MRI negative. And although 7 Tesla has been able to improve this issue, there's still quite a few that are not detectable, even for experienced clinicians. But the one thing is they do exert strong functional effects. So we were thinking about how best to go about things, and what we really wanted to do, just because of the fact that it's going to be quite hard, a lot of the time it's quite hard to detect these FCDs. We wanted to develop a way to get a marker of leptogenicity in the laminar signal. So this is one thing we want to do. So what we wanted to do was, so most laminar fMRI has been conducted in visual cortex. Well, a good deal of it, and it's to study feed forward and feedback connections. So I'm sure everyone in this room, understands, has seen similar things like this. LGM projects the middle layers and then distributed connectivity through visual cortex. So there exist several laminar fMRI experimental paradigms that have targeted different layers to test their role in various aspects of cognition. So one experimental paradigm that I've used before was able to isolate the role of deep layers in predictive feedback processing. So in this study, valid trial started with a color cue that predicted the orientation of a Gabor stimulus with 75% accuracy. And the second Gabor is presented briefly after, and it's clockwise or anti-clockwise from the first. And you signal this with a button press. So the emission trial, so there's another type of trial called emission trials, where the first cue is presented, but nothing is actually presented for the second. So the second Gabor, where they make the judgment, is omitted. And this is the idea of this is to isolate, to control for other top-down influences which might activate other layers. So for instance, working memory in superficial layers, which has been found in a few papers. So here we have on this plot, we have the X. This is results from the study. X-axis orientation, specific Bold signal, and the cortical depth on the X. So when stimuli were presented on the screen, regardless of the cue, all layers were activated. When stimuli were cued but omitted, only deep layers are activated. So we took this as a feedback-driven response in deep layers. So as we've seen in the evidence for the role of deep layers in epilepsy, generalized patients, we want to use this because if we want to isolate a marker, we're going to need to know whereabouts in the brain there's hyperexcitability. So in patients with photosensitive epilepsy, which are seizures triggered by like flashing lights or repetitive stimuli, we know that they have hyperexcitability of the visual cortex. So we know where to look. And does this impact feed forward and feedback activity in deep layers? And this could be used as a marker of altered activity in photosensitive patients versus healthy subjects. And like I say, given the data, we feel this is a plausible starting hypothesis, basically. So one thing is, as I say, detailing where we are with the project, we are waiting for ethical approval to run this part. But what we have been doing in the meantime is we've been developing our methods, and we've recruited a few patients with FCD under another study that does have ethics for us to use their data and to collect a little bit of data. So like I say, we've collected a small number of scanned a small number of patients with suspected lesion. And all we did was, this is actually wrong, it's 7 minutes. We just ran one run of resting state, about 91 volumes. The TR was 3.55, spatial resolution of 0.8 isotropic. And we extracted the data, the pre-processing and data was based on my 2020 paper. We extracted 5 layer depths and we discarded 1 and 5 in the analysis. And so what we did was, working with a clinician, we identified a potential lesion location in functional space. And then we grow a seed in the cortex where we think that is. And then we grow 100 control seed ROIs in the contralateral hemisphere. And this serves as normative data, sort of considered healthy cortex. And then we analyze various aspects of connectivity. So today I'm not going to go too much into this data, even though I've got more than we're going to show, because I'm not 100% sure that it's accurate as yet. And I'll get into that now. But I'm just going to show the type of what I'm going to show is like an example of how we analyze this and what we found. And what we found in this patient that I'm going to show. So this is the median connectivity between different cortical depth in all the control ROIs. So this serves as the baseline. The yellow greens indicate stronger positive correlations. The blues towards the blue indicate weaker or negative. The depths 1 and 2 show strong self-connectivity. Depths 1 and 3 seem to operate more independently. But like I said, this is just sort of an example. I think maybe if I was to come back in 6 months' time, we might have something a bit more convincing. And this is the median absolute deviation, and this highlights how much the connectivity patterns vary across ROIs. Again, higher values mean more variability. Depth 3 is the most stable with the least variability. Depth 2 shows the highest variability. And then what we do here is we subtract the lesion connectivity from the median connectivity. So this is a connectivity in the seed region which we defined as the lesion. And this is connectivity difference. This compares the connectivity in the lesion ROI to the typical connectivity in the 100 control ROIs. The blue indicates a weaker connectivity in the lesioned area compared to the median. And there's a general, in this patient, it's one patient, like I say, I'm not 100% how accurate this is. We still need to work through a few things. There's a general trend of reduced connectivity. And the current issues that we're working on.
Sorry, you're a bit fast. I'm not sure if I got everything. So in these matrices, you're showing kind of the within column connectivity, right? Where you have your RI, and then you look at the correlation of each layer time course with each other layer time course within the same RI, or are we looking at across RI? No, okay. And then you see that within the suspected lesion RI, you're having less connectivity across the layers within that RI, as you would have on any healthy RI on the other side of the cortex. Am I getting this?
Yes.
Okay, okay.
Yes, correct. Yeah. Sorry, Renda, I'm a bit fast there. Yeah.
I'm
Apologies.
slow.
Yeah. And just to say, so a couple of the reasons why I'm a little bit unsure of this data is, so we scan it all in all, working with our colleagues who are doing did quite a substantial project on the use of seven tether in locating FCDs in structural terms. We were managed to tag on a functional run at the end, and we got about 8 or 9. And 4 who had a slightly better defined lesion, they had too much motion, and the ones that had the least motion, we were unsure of the lesion location. And so this is working with an experienced clinician. We are not 100% of the lesion locations for the patients we got usable data from. And then there's other methodological things like ensuring our seeds are actually in grey matter. It's been a bit of a problem. And then we've got ethics. So this is basically where we are. And these are our motivation for the study, what we've done, and where we hope to go next. And then that's it. Thanks for listening.
Yeah, thanks, Fraser. That's pretty cool to see that kind of layer fMRI can be useful for epilepsy, like aside of all the other applications that usually people talk about of kind of investigating physiology or input-output or just feed-forward feedback in quotation marks. So it's cool to
I
have
think
this.
it is.
Yeah. Are there questions? Otherwise, I would start. Yes.
Yeah, I have a question for David. For Fraser or David. Um, so the ethics you referred to, is this an issue of scanning patients with ECOG electrodes or laminar electrodes in the 7T, or is this something else?
It's
just slow.
Yeah, basically, yeah.
Okay.
We've applied for ethics because it's always quite slow to get ethics for clinical. There's quite a lot involved process for scanning clinical children. We're not going to do any sort of IEG. We may do some EEG inside the scanner, but it's just the process of how things go with ethics in basically.
Yeah, so to be super, yeah, to kind of add to that, so yeah, so to be kind of super clear, kind of the aim of the study is to take patients who we know are photosensitive, but to just look at V1 as a photosensitive V1 and look at the layer responses to that task. So basically, we weren't intending on looking in terms of any interaction with possibly ongoing pathological activity. So we weren't necessarily going to record EEG at the same time in those particular patients. Is more just given that they're hyperexcitable, and then based on what Fraser was showing, there's an argument to suggest that we might think that the deep layers are not functioning as we might expect, and so therefore we could use that kind of task to probe whether those deep layers are responding in the same way that we might expect. But yeah, it's just the normal pain of institutions and ethics, not anything specific to the study that's painfully slow.
It's probably taken up quite a lot of my time. That's all. So that's probably why it's in the presentation.
I think Sam's got a question.
And then Lars, yeah, please.
Yeah, that was a really cool talk. Thanks for that. I was just wondering about the practicalities of, I don't know, I guess motion stuff, because yeah, patients obviously move more than our controls, and kids move a lot. And I'm just wondering, like, how much data you guys end up throwing out due to
Yeah, that's a really valid question, because like I say, of the small number of data sets we got, I think we lost nearly 50%. But David has been working on a device called a MinMo. Maybe David, you could just explain what the MinMo is.
Well, I mean, yeah, I can talk, I suppose, yeah, I'm talking a bit more broadly about motion. So I think, yes, it's always the starting point that you take. I mean, that's just true of fMRI to start with, is it's a method that works really well with people who are really compliant and keeping still for cognitive neuroscience. And then you take it into a more clinical domain and it's much more tricky and you have to worry about motion an awful lot more. So I think that's kind of ubiquitous. Obviously, then with layer fMRI, it's another you need another level of motion control. So I guess we are going to be more, very sensitive to motion, and that's always going to be the case. So we are going to throw away effectively a significant amount of data. And probably we won't throw away as much data as you would if you could just go and scan another person, and it doesn't really matter. We are trying to use, so we have a head immobilization device that we've been developing. So we've started to show that can definitely reduce the amount of motion, and in kids as well as adults on the 7T. So that will help a bit. And it's maybe an argument for doing 2D multi-slice EPI as well, where you maybe have slightly better ways of throwing away a bit less data. But yeah, it's always going to be a battle, if you like. And probably you end up with some kids who do really manage to keep still. I think the other factor that we can change is, as Fraser was explaining, so why these, if you're the others, we were bolting on a bit of fMRI data at the end of a study that's aiming to find structural abnormalities in patients with inconclusive 3T MRI. So they've already been in the scanner for quite a long time. And you're kind of fighting that battle, if you like. And that's partly why Fraser's bit hesitant about the results, because those patients are the really tricky ones, and actually what you want is the really clear FCDs where you've got histology on them, because they've been operated and all the rest of it, so you can have a bit more certainty about the location. But all of that just to say, if you have which is the next phase, if you can just dedicate the entire scan session to fMRI, hopefully you get 10 or 15 minutes of really nice clean data at least, which is a slightly different regime to the one we were operating to start with, which is just to try and get a little bit of initial pilot data.
Totally, yeah, super challenging. Thanks.
Lars?
Yeah, I had a question to the task versus non-task. So, first of all, I agree. It's an absolutely fascinating study to see the relevance of correlation between the layers for epilepsy. And you of course, as a motivation, you showed the orientation memory task, but then you can also, you showed that you can do this technique across cortical areas that are or are not involved in the task as well, because they could be also in resting state, be correlated or not correlated. How important is a task, and how much are you thinking about other tasks or just resting state?
Yeah, that's a good question. So the reason we want to use the task first is we want to see if we can, because the FCDs can be anywhere in the cortex, and if they are very radiologically subtle, what we'd actually like to do is use the task to see if we can get this deep layer marker, to see if we can detect this in an epileptic cohort, the photosensitive, and then we can translate this without the task to the resting state where we don't know where the location of the lesion is, essentially. So, and in terms of the task, this is the reason we use that specific task is it's a task I've used before. It may well be changed, but this is sort of exploratory at this stage, and we feel it's a valid task to start with, at least at the initial point, Lars, at least.
Yeah, very nice. Thanks.
Maybe I have a question about second last slide where you talked about the issues that are holding you back. I was maybe surprised to see nothing about methodology. Would you say that kind of the sequence just works, or the analysis is standardized pipelines enough that you can just trust it? Or like there are a lot of method developers in this room who are excited about your study and you have their attention now. What is
well, this is a good one. Maybe I should have talked a bit more about the methods. No, so yeah, there is methodological issues. Yeah. What would you say the biggest methodological issues at the minute are, David? There's a few, isn't there?
Yeah, I don't know about the biggest, but I suppose it's at least we can flag multiple ones in case it piques someone's interest. But so one is obviously the motion you've already highlighted. So that's an issue that we're going to have to deal with and definitely model motion better. And obviously that's going to be layer specific, right? Because I assume that, the superficial and deep layers are going to be more affected by motion because they're near tissue contrast boundaries. So we have to worry about that, I think, a little bit. If we're starting to look across the whole brain, then the quality of our segmentation is also spatially variable. So I think at some point we're going to have to worry about that as well. What are the other things?
Well, I think well at the minute the way we grow the lesions is
Yeah, we've probably done something quite simple to
yeah,
grow
it's quite
the
simple
lesion.
to
So
grow
we
the
pick
lesions.
we sorry, no, is Tyler, you've got your hand up. So did that stimulate a thought or a question?
No, you feel free to finish up your answer. I had a question about your motion mitigation, but I'll wait until after you're done with this.
Yeah, so it's just like, I mean, we basically pick, random locations which should be in the middle of the cortex, and then basically look for adjacent voxels and grow a little region. But as soon as you get into the detail of that, you could easily spend time looking at each one of those and thinking, God, I've accidentally skipped across a sulcus here, or whatever might have happened when you look at the real detail, because if the segmentation wasn't perfect, the boundary wasn't well defined, or all of those kinds of things. And yeah, maybe the last subtle one as well is FCDs often have changes in cortical thickness. So we have to slightly skate over that detail, I think. But what I would go back to is, or the big promise, I guess, the really interesting thing, I think, is, it's a very rare opportunity because you do actually get tissue, which has been looked at under a microscope in some of the patients, not the ones that Fraser's currently scanned so much, as I mentioned before. But if we start to get the patients that have a high chance of being operated, then we can actually look at the cortical disruption under a microscope. And so that's the pot of gold at the end of the quite fraught methodological rainbow.
Will you also be able to record from this tissue extraction? Or just anatomy? Do you also do
I think, well, at the moment, that would just be anatomy. So, yeah, although there are various things that you could do with the tissue in an ideal world, then, yeah, at the moment, it would just be looking at, was this a case with mainly radial or tangential dyslamination, where there are abnormal neurons, those kinds of things. And that can sometimes be spatially variable too. So you could imagine in a larger FCD, you may actually have regions that have slightly different kinds of disruptions. So there's all sorts of fascinating things that you may be able to get to a little bit with that, with, again, the caveat, then you get into the clinical world and you realize that half the time the surgeon's actually just sucked half of it up and just taken a little bit of tissue for the analysis, and you realize you don't actually have half of it. In an ideal world, if you've got a reasonable amount of the tissue, then you could start to look for spatial variation within the FCD, for example.
That's super interesting. I was curious about, so you mentioned very briefly that you are kind of trying to work on motion mitigation strategies right now. I assume is this like a head case situation, or what's the route that you're taking there?
Yeah, so so I'm muted, aren't I? Am I mute? No, I'm
No,
not.
we can hear you.
Sorry, partly because I can hear my children have just arrived home. It's that time of day, so I can hear them shouting in the background. So apologies if you get a bit of that. So we have basically we've developed a device which is basically some inflatables and foam and strategically placed foam pads that we are starting to use. So that we're starting to show does actually reduce motion in kind of scan naive kids. So that's one part of it. I think we will still need to do quite careful modeling of motion as well, and just if we're talking, which is partly the motivation for the task as well, because obviously if we're just doing some kind of resting state connectivity analysis, we're even more exquisitely sensitive to the effects of motion that could kind of trip us up, I think.
Okay.
Whereas at least when you go back to the task case, then you have everything's a little bit more solid, because you hope that your motion isn't always horribly correlated with your task.
And given that I mean, you're dealing with patients that have had sometimes very invasive surgeries, do you find that there are difficulties with like placing padding or whatever it is that you're trying to do to keep them in place, that different people have this problem of kind of you can't really touch the part that you're really interested in, or
Yeah, that's generally not too much of a problem because we tend to be divorced in time from surgery. So we'll typically
okay.
scan people prior to surgery for the focal epilepsy cases. So they may be people who are being considered for epilepsy surgery, but it will be before they actually have surgery. And in the generalized epilepsy case, which we were talking about for the task, then they don't have surgery because there's not a focal abnormality. So yeah, so
Okay,
that's
that
okay in
makes
that
sense.
regard.
I don't know.
Thank you for the great talk. I have a question regarding the scan time and the amount of averaging that you need to do actually.
But how long we scan for, or how long we plan to scan for?
Yeah.
We think for the task we will probably do if when we get clearance to start the task, that will be 2 functional runs of 11 minutes plus an MP2 range, which will be 6 or 7 minutes. So they'll probably be in for about 30 to 35 minutes in total with localizers and this type of thing.
And the resolution that you are using is the current standard 0.8 ISO?
0.8, yeah. And it's slightly less on the MP2 range, about 0.65 on the
Yeah.
MP2 range.
I think you're right. It's kind of an open question is, what do you have
Mm.
sufficient sensitivity?
Mm.
And I think the jury is still very much out.
Mm.
I guess that the hope is that at least in the cases of the FCD, that you're looking at quite an extreme case compared to looking at a subtle difference in this is, so if I was pitching for some money, what I would be telling you is that compared to lamina specific fMRI, normally you're trying to look for kind of subtle changes in the interaction between layers during a normative range. We might hope that actually if you've got a bit of cortex that just doesn't have the right tangential structure or then it's just operating in a slightly different regime. So the hope would be that we have if you like a larger change in signal to look for in the first place. But we'll let you know in a year's time or something like that whether that was the case or not.
Because one of the interesting things is going to be the interictal, because we're not going to be scanning in between seizures. So it's a bit of an open question as to what we would see. So obviously a lot of the animal research is recording seizures, which is a super strong event, whereas interictal it's more subtle. So that would be one thing that's going to be a bit of trial and error, I think.
Maybe I can come back to the results of these like preliminary data that I know you are not absolutely certain about, but I'm trying to understand how they fit in with kind of all the literature that you had. Like you're showing that the layers are doing more different things in these suspected lesion RIs. So does this make sense then that the kind of the deeper layer is basically doing its own thing and it's not really part of the usual column? Or yes, okay. So okay. Or how should I?
Yeah, I think I wouldn't really interpret too much into those just yet. I mean, we see some reduced connectivity, but I'm not confident enough to really read much into that at the moment. I think maybe that would be something that if we come back in a few months' time and we have a few more data sets that I'm confident about, then that would be something we can discuss. But it was just really a proof of concept to show we have this. I don't really like to present anything that I'm not too sure about as a test case.
I understand. Are there any further questions? If not, then I